Hello again. We have a very interesting session now with three speakers. We're going to talk about very different matters, but very interesting. Unfortunately, my co-chair, Benoit Fleury, is not here. So I'm going to chair the session by myself. And Jean-Pierre really makes me work, but it's a pleasure. The first speaker is Mickaël NAASSILA from Amiens, whom you all know, of course. What you may not know is that since is now president of the European Society on Biomedical Research on Alcoholism. He was elected 2 weeks ago in Romania. He's going to talk about psilocybin as a new option for alcohol addiction. Thank you. Thank you. Thank you very much, Karl. Thank you, Jean-Pierre, for your invitation. Thank you, Archives, for the organization. It's really very pleasant to come from the north of France, where it's so cold. It's so warm here. It's a great pleasure. Microphone, okay. It's a great pleasure to come to talk to you at a symposium on alcohol. I took a provocative title, The Return of Psychedelics in Alcohol. We know that psychedelics could be useful in the treatment of alcohol use disorders. So my conflicts of interest, there are a few, okay. As president of the ISBRA, the next ISBRA Congress will take place in Krakow, a beautiful town in Poland, next year. I'm going to go fast because Karl just put pressure. He said you only have 20 minutes, and I thought I would have more time. So I will go fast over some slides. You know how much alcohol is a major issue in terms of mortality, morbidity, global burden, and of course it has a social impact and economic impact. And alcohol is one of the first cause of hospitalization in France. So it shouldn't be forgotten. Karl already talked about treatments this morning and the interest of precision medicine. So with the promising results, there are existing treatments, and now we have to look at the response and see what will what will improve the response. He showed the improved efficiency. The effect of the disease is around 0.4, but we have some efficient treatments in alcohol use disorders, but we need new treatments. The psychedelics, we known for 50 years that studies suggested in the 1950s that psychedelics, and especially LSD, could be interesting in the treatment of alcohol use disorders. With Nixon, we forget about it, and then it's been back in the past 10 years with trials showing that psychedelics such as LSD, but also psilocybin, which is as strong as LSD in terms of number of publications, could be efficient in many pathologies, many disorders, and there are high hopes. To add to what we heard so far, yes, in alcohol, in delirium tremens, there are hallucinations, and why not generate hallucinations in a patient and think that it could improve the prognostic regarding alcohol consumption. As to the mechanisms, what's discussed now is the contribution of psychological effect. compared to the pharmacological effects of the product. That's a major issue. I'm going to show you results with animals, but we don't know about the psychological effects in rats. We don't know whether they're hallucinating or not. But there is a major impact in terms of brain plasticity. You have many reviews showing the interest of psychedelic in many disorder. For alcohol, we have this meta-analysis on LSD. LSD could do better than current treatment, so that's quite interesting in terms of improvement or prevention prevention of relapse. We maintain abstinence with the odd ratio around 2, which is quite good, and the long-term effects if to have a very long-term effect. Of course, they are short-term effects, but not in the long term. Now, I was talking about LSD, but psilocybin, it's interesting for depression, tobacco, and alcohol addiction, with the long-lasting effects. With alcohol, we have 9 months duration. Here you see a 2015 study showing that after a first injection of psilocybin, after 9 months, there is still an effect in terms of decrease on the percentage of days of consumption, of high consumption of alcohol. This is very interesting. There are trials on a limited number of patients. I'm not going to do a review, but there are many studies on what psychedelics in the brain do. Here are 2 studies, the effect of LSD microdose, which will reinforce the connectivity between the amygdala and the frontal gyrus. It has an effect on the mood. And here another study on showing that psilocybin will act on the activity of the sensitive sensory regions or the associative regions. And what's interesting in this study is that connectivity changes were associated with a change in the expression of the genes which code the receptors of 5-HT, 5-HT2A or 1A of serotonin. So roughly the 3 targets of psychedelics are an improvement of the global brain connectivity, an effect on the entropic brain activity, and we expect changes in the neuron excitability. What's very interesting, we looked at psilocybin. So the metabolite is psilocybin. It's a partial agonist of receptor 5-HT2A. It has rapid and long-lasting effects. And what's quite interesting here is that in terms of mechanism, if you know that if you work on schizophrenia, because after activation of the 5-HT2A receptor, there is that could explain long-term effects. You see the mechanism, it has an effect on histone HDAC2, which is going to it will have an effect on the glutamate receptor. What's also very interesting, which I've found out recently with this pharmacology of psychedelics, there will be a presentation on Friday on the subject. They partly act on the 5-HT2A receptor but they form dimers with other receptors and with the glutamate receptor. And here, for instance, in animal, you can lose the pharmacological properties of the psychedelic if you change the gene in the mice. And even stronger here, there is an effect with the D2 receptors of dopamine, and you know the involvement of these receptors in addiction, in alcohol addiction, because the expression of these receptors is associated to a vulnerability to develop addiction. And with addicted patients, we have a strong adaptation of these receptors. On the mGluR2 receptor, we know that there is a reduced expression in the rats. The agonists block the relapse. We can, when we restore the expression of mGluR2 in the frontal, in the accumbens nucleus, we can give back, hand over control of alcohol consumption in rats, and the same in human beings. In alcohol-dependent patients. So finally, the assumption here is that we know that the cortex, as Serge Ahmed said, there are many serotonergic receptors in alcohol addiction. There are less these receptors than mGluR2, and the psychedelics could restore this up-down control by reinforcing the activity of this function. We started working on this project with Rainer Spanier, who is a project leader in an international multicentric test, randomized and controlled in rats in 3 countries, Italy, France, and Germany. The efficiency of psilocybin in a model of alcohol addiction with a collaboration with Roland Weider in Switzerland. We tested psilocybin in alcohol-dependent patient to see the effect on the brain connectivity. So it's a complete translational project. The first results model of alcohol addiction. We can model several criteria of DSM-5. We're quite happy with that. The model we use much in alcohol dependence is upfront self-administration. We also use a period of 8 weeks where the animals are exposed to chambers where they receive alcohol by inhalation. So they have the physical and psychological dependency. They get 2 grams per liter, 14 hours a day for 8 weeks. Then we induce a negative emotional state with a loss of control, different comorbidities. In this model, we tested psilocybin. First in acute withdrawal. What happens? Psilocybin decreases the consumption of alcohol in rats which become alcohol dependent. This will be published next month in Science Advances. In our lab, we looked for the mechanism for this efficiency in psilocybin. The gene that comes out is mGluR2. We have a reduction of this mGluR2 in alcohol-dependent rats. And here, when we treat it, we restore the expression of this mGluR2. We didn't expect it, but this is what happens. We also tested psilocybin in binge drinking. Rats can do that too. They have access only 15 minutes per day, and they will develop high consumption with signs of intoxication. It's a visible intoxication. We just checked that there was no signs of addiction. So it's a pure binge drinking model. We have a liver damage, and they have all signs, non-methane, naltrexone, baclofen, GHB, and acamprosate. So it's a good model in terms of predictivity. I won't go into details, but I show you the most interesting when here we see a decrease in the number of rewards and alcohol consumption. Here, this response over 15 minutes. After an injection, the rat stops consuming. It doesn't explain how psilocybin works. I give you all the results beforehand. It doesn't tell you how psilocybin works, so we decided to inject the psilocybin directly into the brain. And when we inject it in nucleus accumbens, it's the same effect as in a peripheric administration. Here, it's injected in the ventral region. So psilocybin has an effect on the receptors of 5-HT2A. We tested it, and here we block the effect of psilocybin by injected a 5-H2A antagonist directly into the nucleus accumbens. So it goes indeed through these receptors. Even with psilocybin, the animals drink alcohol, and then they stop drinking. What we see that with psilocybin, the rats will stop earlier. We also looked for all the genes that were modified in the prefrontal cortex and nucleus accumbens. We also see that in binge drinkers, we see an increase in the dopamine receptor in the prefrontal cortex. And we know that when you do that, it decreases the consumption of alcohol. So we know that psilocybin is efficient in these models. No modification of receptors D1 or D3. Does psilocybin have long-term effects? So it gets more complex here. After 12 hours after treatment by inhalation, 12 hours later, we still have a small effect. However, 36 hours after treatment, there is no more effect of psilocybin. This is a problem. And there are other tests of psilocybin and LSD in other models, what we call the alcohol deprivation effect, for 9 months at least, with a relapse of consumption after withdrawal. And after LSD or psilocybin treatment, in this model, there is no long-term effect of psychedelics. So it doesn't agree with what we observe in the clinical trials. I will conclude soon. What's developing now is other molecules, other analogs, ibogaine, a psychedelic to develop other molecules that don't have hallucinogenic effects. And this study shows that this analog, a non-hallucinogenic, can block the binge drinking behavior in animals. What's next? There is a project in France to test LSD in alcohol-dependent patients, to test these molecules in preclinical trial, randomized controlled preclinical trials using different animal models. We have to see with the different doses, microdosing, long-term effects on what, on relapse, treatment during the abstinence phase, and then we like imaging in alcohol-dependent patients, because so far I've shown you a healthy patients. I would like to thank all the people, Gregory and Jean-Paul, who did the test on the animals. Thank you for your attention.
Thank you very much, Mickaël. I think it is remarkable how methodology in these clinical trials, how this helps mimic clinical situations. I think this is the way that we have to explore in future randomized studies, double-blind. It's really fascinating. Are there any questions for Mickaël? I would really like to say that this is key, how we can use preclinical trials, randomized and identify respondents and non-respondents. I know this is quite new. And now Maurice, yes, so from a behavioral point of view, how do mice react to psilocybin? Well, we are at a dose where we don't see any behavioral signs. However, if you increase the dose to 10 milligrams, yeah, we can see some behavioral signs, such as the ones shown with ketamine, ataxis or strange behavior, weird behaviors, animals who would just hang from their cages and they do weird things. And what we see in real clinic is that efficacy depends on this initial sensitiveness to the psychedelic effects. So this is something that we should explore much further. We should see more about this initial reactivity. Any other questions? Belance Gadelus, I would like to thank you for your presentation. I'm a clinician, so I would like to know how we can measure in small animals. Well, you know, I'm always quite amazed by these kind of presentations, how to measure the effect. How can you measure whether a mice says, "Oh, I'm high and I've had enough"? You know, this is really my question, because you see that the consumption is reduced, is lowering, but how can you measure this? Do you follow, for instance, the heartbeat, or since mice cannot speak, so how do you know that they've had enough? Yeah, actually, we could use some indexes. For instance, if we are measuring withdrawal symptoms, well, then we see the signs such as anxiety, or there's also a publication on the effect of psilocybin where it shows that there is a decrease in mental flexibility, which means that this molecule is accompanied by the destruction of the inhibitory control. So, well, for us, the parameters that we measure are the classical ones. It's motivational indexes, consumption indexes, seeking the substance, whether we give access to substance or relapse or lack of relapse. And on psychological effects, well, of course, we cannot really go much further on the psychological effects in mice. We measure exploratory activity, but of course, we cannot measure much more than this. So, and this is quite a limitation, because if we don't see any long-term effects, well, then, you know, when you inject psilocybin directly in the brain, we have the pharmacological effect of psilocybin. However, from a psychological point of view, of course, I cannot tell you much more. I would like to know about this reduction of D2 in the accumbens nucleus. What would be the mechanism behind this? And do you observe this only in the accumbens nucleus and not in the striatum, in the dorsal striatum? Or because in other cases, this is especially observed in the dorsal striatum. Well, for us, we have screened everything, and we have screened genes in this frontal area, because this is what's interesting for us. But we saw that psilocybin didn't have any effects, but in the nucleus accumbens, but we didn't look at the dorsal striatum. We would like to see the interaction, and this is something that we need to explore further. We know that there's an opposite communication of this 5-HTA and D2, and I know that we know there is an interaction between both receptors. Okay, really last question. Well, I'm a user and I've used psilocybin, and regarding psilocybin, there are hallucinations. I mean, we're not ourselves. So how can we, how can you make us feel like we remain ourselves? Because when you're on psilocybin, I mean, it's not really like being on LSD, but you know, we do not feel like ourselves. Yes, this is the problem with such clinical trials. We have to manage the administration of psychedelic substances, and we have to lower the dose. We have to really provide low doses. And the dose may vary between individuals. And of course, there's this psychedelic experience of the patient, the trip. So this is undertaken in a controlled environment with a psychologist, a nurse, but this is really the interest of it. Efficacy may depend on this hallucinatory effect, on this psychedelic effect that we have to manage. But somehow, this is the effect that we're seeking to. Okay, thank you very much. And we will now pursue talking about the brain, but now we're going to talk about human brain. And Bruno MILLET is going to tell us about brain stimulation, non-invasive and invasive brain stimulation in alcohol use disorder. And this is going to be so interesting because it seems that this is key in order to move forward in the future. So Bruno, you have the floor. Thank you. Thank you very much to the organizers. Thanks a lot for accepting my communication. This is such a pleasure. I'm going to tell you about some brain stimulation techniques in alcohol use disorder. I had some considerations about the methodological aspects, but I'm going to refer you to a publication. And first of all, I'd like to make you understand why I think it's important to bear all this in mind and why we need to use this invasive brain techniques and non-invasive brain stimulation techniques. So this is the topic, and this is what I would like to present. It's just been published. So it's called Non-invasive and Invasive Brain Stimulation in Alcohol Use Disorder, Critical Review of Selected Human Evidence and Methodological Considerations for Future Research. Because indeed, there is a lot to do and there are lots of methodological limitations regarding these techniques. But, well, this is not what I'm going to tell you about. I'm going to rather focus on the links between obsessive compulsive behavior and the use of substances. Just because I have been working for the last 25 years on obsessive compulsive disorders, OCDs. So I will also insist in the care of alcoholism and stress disorders, and I would also tell you about post-traumatic stress disorder. So the link between stress and the abuse of alcohol and the behavior that we have, this continuous phenomenon. And I'm going to tell you how stimulation techniques may help in substance use disorder, especially alcohol use disorder. And I'm going to tell you about some results. This is our field of work. So first of all, I don't know if you're aware of this. I don't know if you know very well the care for obsessive compulsive disorder. This is basically that, well, when the subject has an obsessive idea, for instance, fear of contamination, and these triggers anxiety, with, of course, all the set of manifestations, physical and psychical and psychic manifestations, and then the individual will feel obliged to perform repetitive behaviors like cleaning or washing. But what's interesting about this is that there are two types or two stages. There's an acute stage, the acquisition of this pathology. So how come that such an idea appears in the mind of that individual, and how this leads to behaviors that will trigger the surge of this obsessive idea? Well, this happens. I can't find the pointer. There's no pointer. Yeah, there is one. Well, anyway, just have a look at the slide and I'll try to describe it. So you can point at the screen here. So the idea is that in the left-hand side brain, there's this anxiety, there's this uncertainty with this directed behavior, directed to a goal. However, this fear, this concern will progressively derive towards a compulsive behavior with the acquisition of a certain habit, a usual behavior. And the brain structures that are involved in this are different, of course. And this is a work that has been performed by a group of experts, actually, where it shows that for individuals having OCD, this OCD becomes compulsive behavior, which becomes quite autonomous. So the obsessive idea is only there to rationalize the behavior, which becomes automatic. So we move from something which is based on a certain reality and on anxiety and concern about the environment, for instance, to a behavior which progressively becomes automatic. And this is translated by a passage in the striatum of a request of a ventral striatum to the dorsal striatum. We need to know that the ventral striatum is very much related to emotional and somehow cognitive aspects, whereas dorsal striatum is in charge of our motricity and behaviors. So these authors are explaining that in the end, there is quite an important modification that takes place. So getting a habit or a behavior is very much explained by this passage from the ventral striatum to the dorsal striatum. And then there's a reward system which depends very much on the learning procedures. All this procedural memory, which leads to this circuit, the corticostriatum thalamostriatum circuit. Yeah, and I'm truly having trouble here with the pointer and the slides. But anyway, so how did I get to addictions? Well, from this OCD analysis, I became interested in substance use, especially alcohol use, because in the same way as OCDs are involved in a vicious circle, it seems to me that it's the same that happens for alcohol use disorder. And I'm not the only one who thinks this, because we see that patients who acquire inappropriate behaviors, well, they do that for OCDs, but also for addictions and for eating disorders and for really severe addictions. It's just as if there was a kind of inclination to this habituation behavior rather than the behavior that is just directed to a goal. And this is confirmed by studies on compulsiveness that can be defined as an excessive behavior or an excessive habit, and also as a lack of control over goal-oriented behavior. And actually, there are studies in rodents that suggest that there's a shift from the central striatum to the sensory motor striatum, with a trend in individuals who use substances. So with a trend for this to be motivated towards a reduction in the control of their actions. So actually what prevails is the ritual, the behavior, the habit behavior over the behavior that would lead them to adapt to the situation. Yeah, it seems that there's the technician trying to help you. Okay, so the idea is to use this compulsivity model adapted to addictions. So it's the idea that the craving would be the equivalent to the obsession for a substance, but there's also the craving that is associated to a high degree of anxiety, and then there's the compulsivity, which really fits the idea of the obsessive idea. So you've got this compulsive behavior, and then there's the role of stress. Stress plays a major role in addictions. I'm going to spare you the stress pathologies in adults, but you must have heard about acute stress, about post-traumatic stress, adaptation disorders to that stress. So on all those patients, the concurrence with addictive pathologies is very frequent. Now, if we have a look at the structures that are involved in post-traumatic stress, we see that we're talking about the same structures here that are involved in addictions with the prefrontal context, also with the HPA axis that plays an important role in the release of hormones that are going to feed this chronicity of stress. So the idea here is to wonder how stress and substance use or alcohol use are related And in this regard, there are certain factors that are related. So there are certain variables, parameters of vulnerability. There is a maladaptive response, which is translated in subjects that are suffering from chronic stress into great reactivity and great sensitivity to the environment, a great difficulty to come back to a baseline. And you will see that in parallel, we often find the use of substances. So it's very much related to this stress-related pathologies. And actually, stress may be the main factor leading to substance use and to becoming dependent. It's one of the 5 factors that contribute to addictions. In addition to genetic vulnerability, you've got social pathologies, you've got trauma and abuse, but you've got stress. So stress favors this fall into addiction. Now, let me tell you now about some brain stimulation tools. I'm sure you've heard about this kind of techniques. There are 2 techniques. There are invasive techniques in which you will implant an electrode in the brain, and the target that is mostly used in psychiatry is a nucleus accumbens. We've just talked about it, and it's a key structure in the reward system. And then there's the subthalamus nucleus that we use a lot for Parkinson's disease patients and OCD patients as well. Now here we can see different experiences telling us that nucleus accumbens is quite promising. This is an area that is quite promising. I'll tell you more about this. But there are also non-invasive techniques. And first of all, we find transcranial magnetic stimulation that allows to have an action on cortical structures quite progressively. However, there's a very low penetration inside the brain, even though with the deep transcranial magnetic stimulation, which is basically about using a helmet, well, it seems that certain structures, such as insula or anterior cingulate cortex, can be targeted. So we're talking about techniques which are promising indeed in order to act upon these systems that we've just mentioned. So in the end, what is the problem? What is the real problem with addicted individuals? I'm going to tell you about what happens with alcohol dependence because this is the clinical part that I know best. But the real problem is decision making. And this is actually what happens. What are we talking about when we talk about delay discounting? Well, this is a spontaneous trend and a very usual trend by individuals to choose a reward. Because in the end, the goal in our life is the motivational system and the reward that we can obtain. So individuals are going to prefer an immediate reward, even though it may be less, rather than a more important reward that will be delayed. So this is what we understand are short sooner or large later. And many patients suffer from this delay discounting, and this really reflects this trait of impulsivity. And we find out that this issue about the capacity by the individual to control this impulsiveness, this is major, and this is what these techniques are targeting. And actually, I cannot tell you much more about this brain stimulation techniques, but let me tell you about some results. because we have found quite interesting outcomes from open label trials. Here we can see some surgical techniques showing that stimulation of nucleus accumbens would lead to a reduction for the craving for alcohol. And we see as well that the deep brain stimulation may lead to lowering of the craving for alcohol addiction. So there are some effects that are correlated with an impact on the dopaminergic system and cortisol system. And well, so how can neuromodulation act? Well, it can act in two different ways. It can help the subject control and tell the brain to control the appetite for the substance, or neuromodulation may target the neurofrontal cortex in order to inhibit the motivation of this reward system. And then we will act on the orbitoventral cortex. Right now, we are performing a randomized trial against placebo on people who are stimulated on the dorsolateral prefrontal cortex with the specificity to act in alcoholic patients suffering from these executive functions. So these subjects are suffering from an important stage of their pathology. And the idea here, as you may understand, is to promote these techniques, which are interesting. And, well, they're just lagging behind a little bit in the field of addictology. And that's why I wanted to tell you about these techniques. This is the team with whom I'm working at the Saint-Pétrière and the Brain Institute, working with addictions, post-traumatic stress disorder, and also OCDs. And this is my lecture for tomorrow or maybe the day after on the use of psychotropic substances. And this is rather for people who want to know about emotional brain and what this means. Thank you very much for your attention. Thank you very much for this tour de force. Thank you very much for telling us about such a complicated and varied topic. Thanks a lot. I would like to go back to the relationship that you established between OCD and addiction, because I don't see this notion of acquisition of the obsessive compulsive disorder. As far as I understood, in compulsion, there is this feeling that the subject feels forced to perform that behavior in a repetitive manner. And then we are relating this to the automatic behavior that you locate at the striatum, dorsal striatum part. But we know that these automatic behaviors are triggered by stimuli which are not necessarily associated with this subjective experience of being constrained or forced that we observed in compulsion. And this is why I really find it difficult to establish this relationship, because actually we become aware that we're performing an automatic behavior afterwards, whereas in compulsion, there is an awareness. The subject feels forced to perform a certain behavior. So that's why I really struggle to see that relationship that you established between compulsive behavior and usual behavior. So this is the first question. And second question, you're telling me that there's a transfer of the nucleus accumbens to the dorsal striatum, and then you're telling me that we are targeting the nucleus accumbens, so the ventral part of the striatum. So why don't you interfere in the dorsal part of the striatum? What is the reasoning reasoning, the therapeutical reasoning behind targeting the nucleus accumbens. If we know that there is a transfer of the behavioral control from the nucleus accumbens to the dorsal striatum. I know these are quite complex questions, but well, thank you. Thank you, first of all, for how thorough you have been with my explanations. And your questions are truly relevant. I do believe that these questions must be addressed. I'm going to start with second question. So why not the coded nucleus, which is the most motor part? It's a very good question. Well, it's simply because today we still have not understood and we have not distinguished the first phases of the disease and the chronicity phase of the disease. And that's why I think that, well, depending on the degree and the stage of the disease in which the subject is, I think that then we have to define whether we target the nucleus accumbens or the ventral or the caudate. And I think that this is a major stake. This is something that requires more reflection. And then, well, your other question is more difficult, and thank you for asking this. I do not have an answer, even though your question is fascinating. Because in compulsivity, do we have this notion of constraint, of being forced to do something? Well, then I would say that at the beginning, the subject feels constrained to do something in response to an obsessive idea which is very present. After a certain time, the subjects report that there's no more constraint. It's just that you have to behave like this. And this is a major problem. And the problem for these people with OCDs is that the environment is telling them there's something wrong about it. I mean, they feel obliged and they know that there's something wrong about it because the environment is telling them so. But I think they're no more in a constrained procedure. But anyway, I think your questions are essential because this is what I would like to draw your attention on. If we accept this comparison with addictions, it will help get some interest in this and develop these targets that are so helpful. I do have a methodological question. In order to get those results that we can use in clinical trials, well, I guess that we need double-blind trials with compared to placebo. So can you tell us about the problem that placebo poses when we're talking about brain stimulation? Because, of course, I think it's quite difficult. Yes, you are truly right. Today, well, I didn't want to get into methodological considerations, but the limitations about these trials on brain simulations is that the sample is really small. And this is the first point. And it's an open label study, and open label trials, they do not have any value today. Which means that we have to go to phase 2, phase 3 trials with large samples, and then we will be facing a lot of heterogeneity, and then we will face also the choice of the procedure and the choice of whether placebo or not placebo. And I'm going to tell you about the transcranial magnetic simulation. The choice of placebo in this is really determining. In most trials, placebo is not a placebo, and so we are actually talking the double blind. And whenever a placebo is a placebo with RTMS, people respond to the placebo. So if we want to distinguish between the good technique and the placebo, we need another stimulation technique that is really strong and efficacious. So, of course, there are lots of methodological considerations to bear in mind.